This Week in Virology, the podcast about viruses, the kind that make you sick.
Esta semana en Virología, el podcast sobre virus del tipo de los que te enferman.
This Week in Virology, episode number 109, recorded on November 9th, 2010. Hi everybody, I'm Vincent Racaniello, and this week we have a special TWiV for you. We're in Mexico City.
Mexico City.
Mexico City, Mexico, and I'm with two old friends, Rosa Maria and Lorena, Ana Lorena. That's the right order? But what's your full name? Rosa Maria?
Del Ángel.
Right.
Núñez de Cáceres. No. Only Rosa del Ángel. Everybody knows.
Not even Rosa Maria?
Rosa Maria del Ángel.
Okay. We'll call you Rosa for today.
Yes.
And your name is Ana Lorena
Gutiérrez.
Gutiérrez. Now wait, we were at a Gutiérrez yesterday.
Tuxtla
Gutiérrez. So, yeah, when I was there, I was thinking about you. But only a little bit.
It's my grandpa place.
Really?
No.
So we're at an institution or a research institute, is that right?
Research Institute.
Called Cinvestav. So
Yes.
what is it? CINVESTAV.
Yes.
What does that stand for?
Centro de Investigación y de Estudios Avanzados. That means Center for Research and Advanced Studies.
Okay.
Huh?
So this is not a university,
No.
right? It's a
It's a
institute.
research institution and also for masters and PhD.
Graduate students.
So you can have graduate students here and postdocs as well.
Yes. Yeah.
Right? And it's not associated with the university at all?
No.
No? And who paid for it? The government. The government?
The government.
How old is this? Is it quite old?
50 years?
It's going to be 50 years.
It's going to be 50 years.
And you
I think 49.
so have you've been here your entire careers in this place, is that right?
I have been here since 1983.
So I should say that I met both of you many years ago because Rosa first came to work in my lab in
87.
So I had only been 5 years in my lab. It was relatively
Yes.
new.
You was really worried about the tenure.
Tenure. I was?
Yes.
You know, I like to think I was never worried about tenure.
No, but you was really worried about it.
Really? I talked about it?
Yes.
I tell people now, I never worried about tenure.
Oh, yes. You was really worried about it.
So I remember your boss at the time was Carlos
Fernandez.
Tomas, right?
Yes.
Yeah.
3 names.
He died.
He did die, but at the time he was alive, and he'd said to you, you should come and do some work in my lab? Is that
I
right?
decided to go.
Oh, you decided. How did you
Because-
know where to come? How did you know? Did you know our work? You read about it?
Yes.
No.
Your papers.
So you decided to come.
And I wanted to express structural proteins, poliovirus structural proteins. And in my lab, it doesn't work. It didn't work.
Okay.
And I wrote a letter.
A physical paper letter.
Yeah.
Yeah, this was before email, right?
Yes. And you never answer me.
No?
No.
I'm sorry.
But you
The
never
truth
answer.
has to come out sometimes.
You never answer. And I call you.
You on the telephone?
Yes.
Amazing. This is before cell phone,
Yeah.
before email. Oh, that's amazing. Okay.
And you told
called
me, okay, come. But I don't have money to pay you.
I did say that?
Yes.
Yeah. Okay.
And I get money. I got some money. And I went to New York to work to your lab.
So at the time you were a PhD student, right? Here?
Master. I'm finishing the Master in Sciences too.
Okay.
Degree. And I think I only worked for a week and I finished my Master in Science.
In my lab.
In your lab.
But you stayed longer, right? And
Yes,
did some other work.
and I started 2 projects.
Right.
One with SFP8 mutant. Do you remember the double mutant in the
Of a polio
5'
vaccine strain, right?
non-coding
Do you remember
region?
too? That,
Yeah.
Wow.
Sure.
And the other one was proteins binding to the 5'
Mm-hmm.
non-coding region, yeah. And that we published in PNAS.
Mm-hmm.
It was a good paper. So then, how long did you spend? A year or less? In my lab? In my
Less, I think. The first time less, and the second probably 6 months, something.
So you the first time, then you came back here, you finished your PhD?
No, the master, and I start PhD,
Yeah.
and I went to New York again.
This was a year later or more than a year later?
No, I think less than a year
Okay.
later.
And then you finished most of your PhD work
Yes.
as well.
Mm-hmm.
And then that was still in the 1980s, right?
89.
Lorena, you had not come yet.
No,
No.
until 93.
So you came back here and started a lab of your own here.
Yeah.
Yeah.
And by then your advisor had passed away, Carlos,
Yes.
right? And so was this the lab you're in now? Was this his lab?
No.
Was it another
In
lab?
another department.
And then you started having students, and Lorena was one of her
I was-
students.
yeah.
First
No.
No, I was
couple.
her first PhD.
Mm-hmm.
Yeah.
And then you came to visit. So Rosa suggested to Lorena that you should come to my lab.
Yeah, I wanted to also.
Oh, you wanted to
yeah,
also?
she should suggested, but I wanted to do also.
And then you came in the 90s sometime?
93.
For how long?
It was like a couple of months, I think,
Okay.
3 or 4 months.
And at that time you came alone, right?
Yeah,
And then you came back here and finished your PhD.
I was just starting the
Or
PhD.
just PhD. And then that was it. For Rosa, you only came twice, right? Or no, you came later to write a paper.
Yes.
I went with Rosa to write a paper. So
That
I
was
was
much
taking
later,
care of
yeah.
Aiden while you were writing.
Yeah, so we should say you had both a lot of data. And in the meantime, another student came with you once, Esther?
Iris.
Did she come with you or by herself?
No,
With-
she was there when I arrived. She was there like for 1 year or 6 months.
1 year.
No, 1 year,
Okay.
yeah. And she
And-
was already there when I...
And then she left.
Okay.
You remained. So were you in my lab twice,
Yeah.
Lorena?
No,
What do you
just
want me to
once.
call you, Lorena or Anna?
No, whatever it's easier for you.
Okay.
That's fine. Anna, it's okay.
So you were in my lab a couple of times both, and then I remember we had a lot of data, and you kept, by then we had email, and you said, we have to write a paper, and of course I never did anything. So finally I said, you should come, and you both came for a week,
Yeah,
and we wrote 2 or 3 papers,
Mm-hmm.
right?
2 papers.
2 papers.
So now both of you have labs here of your own. You're professors, right?
Mhm.
Yeah.
And you're doing independent research.
Mm-hmm.
So the point is that we have known each other for a long time. You spent time in my lab. I even visited here once, right? 20 years
20
ago to
years
give a
ago.
seminar. So now I'm here because we were at a meeting in Tuxtla Gutiérrez, which
Mm-hmm.
is the same as your last name. And then we came here and I just gave a seminar here.
Okay.
And it's really great to see both of you again. You're like old friends. It's too bad it was 20 years that, yeah. I've seen you at meetings though over the
Montreal
years.
and Davis and
So I want to know, I'm always amazed that you can be scientists here in Mexico and do virology. So I want to talk a little bit about that. But I want to know, when you were growing up, when did you get interested in science? Rosa, I mean, in high school or before?
I think in high school
You liked science.
I like science, and when I finished the degree, the college,
Degree, yeah.
I came to Simba
Yeah.
and I met Carlos Fernandez, and he told me about the work with poliovirus.
So you met him at college? No, you met
No,
him
here.
So why did you
Here.
come here to Sinvestab?
Because a friend of mine told me that here people make good science.
I see. It's always word of mouth that's important. Someone tells you, right?
Somebody told me. And I came first to the biotechnology department and later to genetics and molecular biology department. And I preferred to be in the genetics and molecular biology department. And there I met Carlos, and he told me about his work with poliovirus.
Right.
And I was, I studied food technology.
Food technology.
Food technology.
Right.
But
This is important.
Food technology,
not microbiology, but technology.
I studied chemistry, but in food technology.
Okay.
And that's why I decided to come to biotechnology department.
I see.
Because in that time, people was trying to make on a cellular protein or some other things. But genetics and molecular biology department was, I think it was a better option for me.
Yeah.
And I like viruses. I don't know anything about viruses.
Now, why did you like them? What did you hear about viruses? Like in school, were you taught virology at all?
A little bit.
I see.
About viruses.
It's useful to point out that Carlos worked for David Baltimore for a while, right?
Yeah, as a postdoc.
As a postdoc?
Yes.
So I knew his name when you contacted me initially. So yeah, and this is another way that science works. People know each other from different labs, right? And say, okay, well, he was in David's lab, so you must be okay. So yeah, you can come to my lab, right?
But you make a mistake.
No, I didn't make any mistake because you were both doing really well, so no one can believe that. So you came and started working for him, and that got you into the master's and PhD
and the
program.
PhD.
What about you, Lorena? You also liked science very young age?
Very young age, yeah. As I remember, in high school, I have a really good biology professor.
Yeah.
And then I get in love with virology.
Yeah.
Yeah, and then, well, I decided to study biology. And then during the last days of the career,
right.
So I just have Carlos Fernandez as the virology professor. And he was a great teacher. So because I met him there, he just told us that we could come and do the thesis research here.
Right.
So I was in his department, not with him because he was full. His lab was full there. So I just did other things. And finally, when I got the degree, I started looking for master's and PhD. Yeah.
So was your family scientifically oriented at all?
No, not at all.
How about yours, Rosa?
Not at all.
No.
It's interesting because sometimes it comes from the family, but that's good. That you can come to science from not having any influence at all. Maybe sometimes when science is in the family, the kids don't go into science like my kids probably won't because we're both scientists. But that's interesting. And then you came here, you both did degrees. And then, so when you get a PhD in Mexico, do you do a postdoc often or no?
It's good if you make a postdoc.
Yeah.
But in that time, since I have been working in your lab, I think it was not necessary. And I got a position here, and I think it can be a good option to stay here.
Is it hard to get a position here? Here in Mexico in general? Is it
in that time, it was not so hard. In this moment, it's really difficult.
Yes. Yeah. And in these days, you need a postdoc, to have some publications in order to get a job.
You didn't do a postdoc either, right?
No, I didn't.
Because you worked in my lab, it was okay?
Yeah, because
I can't imagine that that's true.
No, it wasn't that. Because Rosa was like a independent professor.
Yes.
And then when I finished the degree, it was okay that I was in your lab. But I started as an associated professor with her. So it was easier that way.
Yeah. Now you have your own lab, right?
Yeah.
You work independently. You're both independent. You have you both have students and postdocs. So I did a good job if I did anything.
Yes, of course.
I didn't do anything, but at least
you did a lot.
I gave you my feeling is I give people an environment to work in that's good, and that's all.
You don't even give you You give me your own place in the lab.
I did? I gave you my bench, yeah?
Yes, your bench, but it was hard, because you didn't want to.
But I did. There was no room, so
and
it was
you
fine.
Remember that Alan Dove was in the middle of Iris and Ice, so we called him that he was in the Alamo.
That's great. I remember. So Alan Dove may listen to this. If not, we'll tell him. But he's, of course, on TWiV a lot.
Yes.
And he will remember you, Lorena, and you as well, Rosa? Do you remember Alan Dove?
I don't think so.
You were before him.
Yeah.
Yes.
Right? So in the U.S., women in science is very tough. It's really, we're always trying to get more women in science. My department has a couple of female scientists. So I'm amazed that here I have two women who came to my lab, and they have independent positions. They're doing good work. But I get the feeling, and you should correct me, in general in Mexico, maybe it's better for female scientists here than in the U.S. What do you think about that?
And I think it's possible.
Yeah.
Because I think the competition in the U.S. is harder than in Mexico.
Mm-hmm. So what's the fraction in a science field in Mexico? How many women versus men? You have any? It's not 50%, right?
No, I don't think it's 50%.
No.
I think it's probably 70, 30.
35.
So this, are you in a department in this institute? It's
Yes.
called,
Yes.
what is it called?
Department.
Of what?
Infectomics
This
and molecular pathology.
Infectomics and molecular
Yes.
pathology. And
Our name was Patología experimental pathology.
still on the
But
door,
now-
I think. Yeah.
Yes.
But now it's infectomics
They changed it. Okay.
and molecular pathogenesis.
Oh yes, that's right.
So how many professors are in this department?
16.
16.
And how many women?
Oh, let's guess. Yeah, like 7 or
7.
8.
8.
8 out of 16.
Yes.
50%.
50%.
It's amazing.
Yes, but in other departments
It's less.
it's not the
Okay.
same.
So it may be that there's less competition, but there is competition. So
Yes.
you should not minimize what you have done.
No,
At
no,
all.
no. And you have to publish, and you have to have students and everything. But probably in the US, the competition is harder.
Right. And both of you have kids.
Yeah.
So it's even harder,
Yeah.
Yes.
right? So how do you do it?
We have good husbands, I
Yeah.
think. Because you need a good husband to make science.
Some people think that the words good and husband never go together.
No, they're wrong.
You have good. So you agree with that?
Yeah, for sure.
So what does it mean to be good? They support you in whatever you want to do?
And help you.
Yeah.
And if you have to travel, they help with the kids. And I think it's important to have someone who help you
Yeah.
and who
Support you.
understand you.
So you're lucky, right?
Yes,
Yes.
absolutely.
So you've had good support to give you time to put into this work, which is what you need to be successful, right?
Mm.
Are there a lot of young women in Mexico now trying to go into science?
Yes, I think so.
Do you train them in your labs as well?
Yes. Mm-hmm,
And does the future look good for them, or is it going to be very difficult?
Well, as in life, some of them just look very
Yeah.
good,
Yeah.
and some others not as good. But I think that they will do really fine. Isn't it?
Yes.
So
Most of them, yeah.
what kind of advice would you give to a woman in Mexico who wants to be a scientist? What's the most important thing she can do?
Work hard.
Always goes without saying, right?
Study.
Stay focused.
Stay focused. And one thing is, don't get stressed about experiments and that, because sometimes, this is very difficult when you get wrong results and that you finally get a little bit disappointed of that. So you have to keep on doing that and do it again and again. And you have to be really sure that there are times that you have to work harder and others that you don't so hard. You always have to work hard, but you have to you have your space in order to
Sure.
to do the science things work better.
You have
So
to-
I don't know if you understand what I
I
meant.
understand. And you have to balance your
Exactly.
family too, because they're
Yeah.
always pulling for your time.
And if there is not a balance, you're not going to do anything. Not in the science field, not in your
Personal life.
motherhood or
Yeah.
And also, I think one important thing is to collaborate. I think in Mexico, it's not so easy to have a good collaboration with another professor.
In Mexico.
In Mexico.
Why? Why is that?
I don't know. I think it's. I really don't know. But the people don't like to collaborate. But I think it's very important, because it is impossible in this moment that you can do everything. It's impossible that you can be an expert in immunology, in molecular biology,
Sure.
in genetics. And I think the best thing that you can do is to collaborate with other person that can help you with a part of your research.
Yeah, it's always a problem in the U.S. as well. I mean, collaboration makes things happen quicker, but scientists like to have everything
Yes.
for themselves.
Under control.
Big
Well-
egos.
Yes.
But I think that sometimes it's like that, sometimes not. I wouldn't be so radical on that. No, I agree with you, but you can find people that help you and that collaborate with you. Nice.
So have you had collaborations within Mexico that work
Few
for you?
because there is no too much people working in my field.
So virology in Mexico is not a big field, you would say?
Yes, it's
Really?
not. It's not
not
yet,
a big-
but it's becoming.
Last year, of course, Mexico got a lot of attention for virology,
Yes.
but that did not help to make virology more prominent.
Yes, I
Must have,
think
right?
it helped, and I think that there are now much people doing virology.
Yeah.
Not as we would like to, but I think it's increasing.
Yes.
Isn't it? And there are really nice persons doing that. Not nice persons, but
Sure.
people that is doing it well.
Mm-hmm. And I think we have very good opportunities to make virology, because virology is a very important science in this time. Because in Mexico and as in other countries, we have very important diseases caused by viruses. And in Mexico, there are really few groups working with HIV, for example.
Mm-hmm.
And this is a very important pathogen
In Mexico.
in Mexico.
Yeah. Mm-hmm.
And there are really, I don't know, probably 2 or 3 groups working with HIV,
Yeah.
and mainly in a clinical research.
Sure. So HIV is important. Influenza, obviously.
Hepatitis C, nobody's working with hepatitis C.
Is it
in
a
Mexico.
significant problem in?
Yes.
Yes.
Yeah.
Yes. As in other countries.
No one is, very few, or no one is working on Hep C?
No one.
That's amazing.
No one is working in hepatitis C.
So future
Some groups are working in hepatitis
virologists should work
C.
on Hep C for sure. Hep B, is Hep B a problem here? It is
Yes,
everywhere
yes.
else.
Yes.
Yes.
Dengue, of course.
Dengue is an important infection.
What else? Anything else that
Nobody's working with hantavirus.
are those important here in Mexico?
I don't know.
I don't know. We don't have a very good epidemiological studies.
I see.
Only some infection are studied.
So is there good support for research here in terms of money? Is it easy or is it very difficult to get support for your research?
Sometimes it's easier. If you are a professor with good papers and good amount of students, it's easier to get money. If you are a new professor or new researcher, it's not so easy. Because in Mexico, you only have CONACYT grants.
CONACYT, CONACYT.
Yes.
It's like the equivalent of NIH in the U.S.?
More or less, yes.
More or less. It's an organization that give grants
Right.
for
Science
and technology. And I think in Mexico is the only, and now the Instituto de Ciencia y Tecnología, Science and
Mexico
Technology
City.
of
It's
Mexico
particular
City.
for solving Mexico City problems, but
I see.
they are giving
So
grants.
is the main research in Mexico City, or are there other cities that also
There are
have?
other cities.
There are other cities, but I would say that the main research is here. There are other important cities
Yeah.
also.
I know that in Cuernavaca there's an institute
Yeah.
Yes.
where Susana Lopez
Yes, and
and
Carlos Arias.
are
Yeah.
both.
In Simbestap, but in
Yucatan.
Irapuato,
Mhm.
there are some groups working with plant viruses.
Okay.
Also important groups.
Agriculturally important,
Agriculture,
right? Yeah.
important viruses.
Because you have a big agricultural industry here, right?
Yes, an important industry.
Or anywhere else in Mexico?
Yeah, in Yucatan,
Yucatan?
Guadalajara, Monterrey.
Okay. So one thing that I always wondered about, so there's a lot in this country that requires money.
Yes.
There are a lot of poor areas that could use help. How do you decide what to do with the money, research versus helping people? Do you think about that issue at all? Because I certainly have, looking from the outside.
Yeah.
Because they say, oh, you spend this much money on research, but maybe you should help people who are less well off.
Yeah.
Does that come up?
Yeah,
Mhm.
but I think that you have to fix both things.
Yeah.
Because poverty can be fixed if you just give the money to hospitals or things that are really an important issue for a lot of people that doesn't have that.
Right.
But in the other side, you also need to give money to make basic science. Otherwise, the country will not be independent of
Yeah.
that, you know.
Mhm.
Yeah.
So maybe it's not a half and half, you know. Maybe you should give much more money to the basic necessities. Is
Right.
that correct?
Right.
But you cannot forget about the other side of the coin, because it is important
So
to the developers.
what do you think, Rosa?
I think you need to use money for science, and also to use money for solve our problems. Include money, food, jobs, and everything. The problem is people is not always see the results of the money give
To science.
to
sure.
science.
It takes a long time.
because
right?
it takes a long time.
Yeah.
People said if you are working with the vaccine, probably you will have the vaccine in 1 year, but it doesn't happen in that way. You need
Sure.
long time to see the results, and sometimes it's not easy to see the results as soon as you
I
want.
guess Mexico is lucky because you have money from oil industry, so you have money to spend on research. There are many countries that can't even do that because the small amount of money they have has to go towards helping people. And of course, many countries have corrupt governments where they take all the money themselves, so there's nothing for science. So you're lucky in a way that you can do that, right?
Mhm. Yes.
Are there enough young people interested in science to keep it healthy going into the future?
Yes.
Yeah?
I think so, yes.
And you see some of them here, obviously.
Yes,
right? Sure.
Absolutely.
I think it's great that you can do this, that you have established really nice groups. I'd like to talk about what you're doing here a little bit to see what's going on here. Now, when you both did polio projects in my lab, right?
Yes.
But you can't work on polio here,
No,
right?
No.
So what they said to you, you could do it as a PhD thesis, but if you want to have a lab, no polio?
we worked with poliovirus some years.
Yeah.
However, in I don't know how many years ago.
Not like 4 years ago.
No, I think more.
Because
Government
I was already in my...
Okay. Government decide to eliminate all poliovirus in,
Really?
in all laboratories.
Oh, for the safety
was
reasons.
because of
Yes.
the
Eradication program.
So here polio has been eradicated. The whole North and South Americas, it's eradicated, right? So you still immunize here
Yes.
for polio
Yes.
using the Sabin vaccines?
Yes.
Right?
Yes.
So in the U.S., we switched to the...
Now some programs have also Salk, but I think they still giving the
In Mexico
Sabin
is Sabin vaccine. And they come to our lab and destroy all poliovirus strains, your poliovirus strains, and destroy everything.
I knew then I felt some pain. I didn't know what it was. So for the safety reason, they said you have to destroy everything.
Yes.
And then you had to find something else to work on.
We start to work with dengue probably in 94, 93, I think.
Mm-hmm.
And it was easy for me to change because some of my students are working with dengue and some others with poliovirus. And dengue is an important problem in Mexico.
How important is it? In the U.S., it's not a problem yet, but why is it a problem
in Mexico,
here?
it's an important problem. We have dengue in almost, I think, 29 from 32 states.
Mm-hmm.
We have outbreaks in almost all states. The mosquito is present in almost all the Mexican
Which mosquito
republic.
is transmitting it?
Aedes aegypti.
Aegypti. Okay.
And also, we have the 4 serotypes co-circulating.
Ah.
And this is a big problem.
Do you know how many cases a year, roughly?
Last year, we had, I think, 45,000 cases.
And this is the hemorrhagic fever, not the shock?
No. This is normal cases, dengue, classical dengue fever. And hemorrhagic fever was, I think, 9,000 cases. And the problem is death.
How many deaths were there, do you know?
In this year, we have, I think, 25. And people is worried always because at the end of the raining season, the number of cases increases. I think in this moment we are having a number of cases, an important number of cases of dengue.
Is it a seasonal disease here, or is it all year?
In the south part of the country, it happens all year, but in the north part, it's a seasonal problem.
And in the north, of course, then it goes over the border into Texas in the U.S., right?
Yeah.
Because the mosquitoes don't pay attention to the border, right? They can just go over, and the virus probably goes back and forth.
Yes.
Eventually, it will spread to the U.S. where the mosquito has a range as well.
Yes,
So-
but I think in U.S. you have another Aedes mosquito, probably
Albopictus, yeah. I heard that it's not as good a host for dengue as Egypti.
It's better host Egypti.
Yeah, but Egypti doesn't have the range in the U.S. as Albopictus. Albopictus
Yes.
is pretty much everywhere.
Yes.
So you can get funding in Mexico because it's an important medical problem.
Yes.
And I guess the goals are to make vaccines one day, right?
I hope so.
So, but that's not what you work on, right?
No. We are not working.
So I want to ask you just one question about vaccines. So right now, if you are infected with serotype 1 and you have mild disease, you recover, then you have another serotype and you can die because of the shock syndrome, right? So if you have a vaccine and you have immunity to all 4 serotypes, aren't you going to get shock syndrome then, more likely?
No, I think if you have enough antibodies, neutralizing antibodies against the 4 serotypes, and you can have dengue hemorrhagic fever.
Okay, so it will block infection,
Yes,
so there's no
you can
chance of
block it.
having the serious
Yes.
disease. And there's some candidates being clinic tested globally somewhere right now, right?
Yes.
And maybe these could be used in Mexico someday. Is that the goal?
Yes.
So you have to be careful. If they eradicate dengue, or at least control it, then maybe you can't work on it anymore, right?
I hope no.
I mean, if there's a vaccine that prevents, it can't be eradicated, probably.
No, I think it's very
right?
difficult
Because there
to
is
eradicate it.
there's a monkey host as well, right? No?
It's not clear that the monkey is a good host. Because we have been testing monkeys in Mexico, and
These must be New World monkeys, right?
yes, probably it is possible. I don't know.
And you don't find
No,
dengue in them?
No.
Because I've read in the textbooks that there are some cycles.
Selvatic.
Sylvan dengue, is it? There's a cycle between monkeys and mosquitoes in the forest,
Yes.
and then there's human dengue.
Selvatic?
Sylvatic is in the forest, and what's the human? The human is urban dengue, I guess. And the question is whether they can go back and forth, and there's some debate about that. So you can imagine that if you eradicated human dengue, maybe someday the monkey dengue would jump back in. But let's say you could control it with a vaccine. Lorena's doing work now. You're not
I'm not
interested.
doing work.
You're not doing work? You're reviewing a manuscript. Would you have to stop working on it, or the government says, well, work on something else, or maybe that won't happen in your career?
No, I don't know. I think dengue will be a
problem for a long
problem
time.
for a long time.
I think it's not going to be easy to get a good vaccine that works.
No.
So what's your interest in dengue? What problems do you work on? I heard you give a seminar yesterday in Spanish.
In Spanish.
It's and the slides are in Spanish too. So all I remember is you're working on the severe disease, how the virus might do that, right?
Yes, we have, I think, two different lines in the laboratory. One is involved in entry, and we are analyzing changes in the cytoskeleton, also signaling pathways activated after infection, and also molecules involved in viral entry and vesicles involved in viral entry. And also the function of the role of cholesterol in this process. And in another way, we are also working with severe disease and trying to find some markers in patients, some markers between dengue fever patients and dengue hemorrhagic fever. And we are looking for some cytokines, are also the presence of the non-structural protein NS1 in serum from those patients. And also some other cytokines and TNF-alpha factor and some other molecules.
Mm-hmm.
And this is the light side. In my laboratory, people said that we have two sides. One is the light side. This is the light side. And the other is the dark side. The dark side is about the RNA molecule, replication process.
Oh, that's the good stuff.
Yes.
Yeah.
I'm always in
But
the
this
dark
is the dark side.
Great.
This is the dark side. And in this case, we are working with some molecules that interact with viral RNA. And we have found some molecules such as PTB, LA, and some others like EF1 alpha, calreticulin, PDI. And we have found some that proteins such as LA and PTB are regulators, LA as negative regulator, while PTB is a positive regulator of dengue replication.
Right.
But we are trying to find the function of some other proteins such as calreticulin and PDI that both are chaperones in the endoplasmic reticulum. We don't know if these proteins are required for movement of the RNA from the vesicles
Mm-hmm.
to the endoplasmic reticulum, or if they are involved during replication process, or if they are required for viral morphogenesis. And we are trying to analyze these aspects. And also the role of cholesterol in the endoplasmic reticulum during viral replication.
So cholesterol is important both for entry and
for replication.
maybe for replication.
Yes.
In replication, does it have to do with making vesicles that the virus needs to replicate its genome?
Yes, and possible because dengue virus uses like vesicles inside the endoplasmic reticulum
Mm-hmm.
to replicate the genome. And it's possible that these vesicles and the new membranes
Mm-hmm.
generated after infection
Right.
could require cholesterol.
So the genome is replicated in the endoplasmic reticulum?
Yes.
And how does it get out then into new virus particles or mRNAs? Say,
By budding
from the ER into the cytoplasm?
Yes.
Yeah. And mRNAs are made in the ER as well?
Yes.
And they bud into the cytoplasm?
No, yes, to the cytoplasm, they go to Golgi apparatus.
Golgi, right?
And then through secretory pathway.
I see. Okay. And if you deplete cholesterol, it interferes with this.
Yes.
So what we need to do is take cholesterol out of our diets, right? Is it possible?
We have used some drugs like
I see.
statins and some others, but people from Novartis
Mm-hmm.
have used some siRNAs to silence the expression of some enzymes required for cholesterol synthesis, and all these siRNAs reduce viral replication.
So the idea one day maybe you could have an inhibitor of this process, but we need cholesterol, obviously.
Yes.
But maybe you could make specific inhibitors that would inhibit viral replication.
And I think it can be they can be used only for a short time.
Right.
And try to avoid the complications, dengue hemorrhagic fever or shock syndrome.
So if someone has dengue, how do you tell? Is there a diagnostic test?
Yes. There are very good diagnostic tests because people There is protein called NS1. This is a non-structural protein, and this protein is secreted from infected cells.
Okay.
And you can find it in the serum from patients, and you can detect it from day 0. If you say 0, the day that you have fever,
right?
The first day you have fever, from this day to day 6, 7, you can detect NS1 in the serum.
So is that used for diagnosis?
Yes.
NS1? So was it an ELISA or some kind of antigen?
There is an ELISA and the other tests similar to the...
Yeah, they're done on a dipstick or
Yes.
an antigen kind of
Yes.
capture test. Yeah.
Yes.
So this is used to diagnose.
And it's very sensitive and specific because it is only specific for dengue.
So right now, so is dengue diagnosed, and then what can you do? Nothing, right?
Nothing.
You just make sure
If a patient is only with symptoms like classical dengue fever, you can give the patient paracetamol. I don't know if it's the same name in the US.
Sure. Yes, it's Advil or...
Advil.
Yeah.
Yeah, so you just take care, make sure they have no fever and that.
And that's it.
But in the future, if you had an antiviral, you could give it to them. Since you have a diagnostic.
You have to be really sure that the patient is okay, because if the patient have some hemorrhagic symptoms, it's better to keep it in the hospital.
Sure. We talked before about animal models for studying the disease, which would really help to understand. So you have ideas about cytokines that might be involved in serious disease, but having an animal to study that would be good.
Yeah.
Is there a good animal model for dengue, in your opinion?
Eva Harris and Rebecca Rico have very good models, I think. I think the best models we have in this moment, they have the best models.
Yeah.
But it's not exactly the same as happen in humans.
We have talked about dengue quite a bit on TWiV, and there are, from what I could tell, there are some mouse models where you infect and you have the normal dengue, and then there are other models where you infect and then you give the mice antibodies to a different serotype and it makes the serious disease, right? So they're all artificial, obviously, but they allow you to study certain things. But you don't work with mice at all, right?
No.
Do you want to, or is that you're the dark side?
I could like to do that. No, I could like to do that, but in this case, I'm working with patients and try to find something.
It's actually a good. I mean, I think that if you can learn something from studying patients with the disease, if you can have correlations in certain cytokines, or that might give you more information than a mouse, right? Because mice are mice; they're not people, and sometimes they're very different.
Yes,
as we've learned in many examples. Do we know the receptor for dengue, or is there more than one? It's controversial, right?
It's controversial. It has been described several molecules. In monocytes, the DC-sign. DC-sign is a C-lectin present on the surface of dendritic cells. And this molecule interacts with dengue virus and also recognize the mannose receptor, the mannose residues present in the surface of the E molecule.
Right.
The problem is, I think this molecule is only required for attachment, because if you remove or delete the domains, the intracellular domains required for endocytosis, it doesn't happen. It doesn't happen.
Yeah.
The virus enter into the cells. Then it's not necessary the molecule or the interaction with this sign to enter into the cell. It's only required for attachment.
Then there's something else that brings it in the cell, probably, that hasn't been identified.
No. Another receptor is the mannose receptor in monocytes. Another group have described a laminin receptor in Hep G2 cells, and also GP78 is a heat shock protein. And also our group, we described two molecules, HSP70 and HSP90, as putative receptors for dengue virus.
I remember.
But it is not clear if which of the molecules or all the molecules, or the virus uses different molecules in different cells, or you need to interact first with one molecule and then with another. Heparan sulfate, it is also important for the interaction. It's not clear which molecule is the receptor.
We're lucky with polio.
Yes.
There's one molecule, and it's the same in every cell.
Yes, and absolutely. And if you have seen, for rotavirus, it's the same problem. There are sialic acid and also some integrins and also some heat shock proteins.
Right.
And it's not clear which molecules are really required for viral infection.
But some mice can be infected with certain strains of dengue. So whatever the receptor is, it must also, some of them must be present in mice as well.
And in vitro, dengue is a very promiscuous
It infects
virus.
a lot of different cells.
Infect almost all type of cells. That means that probably the receptor, or the virus can use several receptors,
probably can,
or the receptor is present in a lot of types.
Are you still working on understanding the receptor? Or is that no?
It's not so easy.
Yeah, I know. It sounds well. Many people have tried, and as you said, it's confusing.
It's confusing, and I prefer to study the mechanism used for entry, the molecule requires during the process.
And you talked yesterday about how there's a model where antibodies, non-neutralizing antibodies, will bind the virus and bring it into monocytes via FC receptor. Is this still what the field considers to be how serious disease occurs?
I think this is the main risk factor for dengue hemorrhagic fever. But I think there are some cases in which in a primary infection, people is really sick, probably with hemorrhagic fever.
Yeah.
And sometimes, and in 90% of the cases, if you have a secondary infection, you don't have dengue hemorrhagic fever. That means another factors are probably involved in the process.
So I don't know if you know, but for both influenza and HIV now, a really interesting development is the identification of antibodies that can neutralize many different strains.
Mm-hmm.
So for HIV and flu, they are directed against the stem of the HA. And not the head. The head makes very strain-specific neutralizers, but if you can direct them towards the stem, and the same with HIV. So the prospect is now with influenza, maybe at some point you could immunize at a young age with one immunogen and protect you forever against any strain that could emerge. So does anyone in the dengue field think similarly that maybe there are some very conserved epitopes among all four serotypes that could be used so you avoid this problem of, non-neutralizing antibodies enhancing disease? Is this an idea or not at all?
No, not at all.
No.
I think not at all. But
it could be interesting
to analyze.
Because for flu, I mean, the problem is for flu and HIV, they're such big infectors that you get a lot of attention, right? So what they did was to identify for HIV patients with antibodies that can neutralize many different strains, and then they cloned the B cells from them and cloned the antibody molecules, the DNAs, and said, these are antibodies that can neutralize many strains. So this might be something to do for dengue.
Yes, and in the case of dengue, there are also other antibodies that can increase the susceptibility to have hemorrhagic fever. The antibodies against PRM, the precursor of the membrane protein. This protein is present only in immature viral particles, and they are not, under normal conditions, they are not infective. However, if you use antibodies against this molecule, the non-infective virus can be infective. And it had been demonstrated by Yolanda Smith. And also antibodies against NS1 can recognize endothelium and increase vascular permeability, or can recognize blood, how do you say? Plaquetas.
Platelets.
Platelets.
Platelets, yes.
Platelets. And inhibit aggregation.
So you have some self-recognizing autoreactive antibodies, basically, right? They cross-reacting with cellular components.
Yes.
It's a problem, right?
Yes, it's a.
Do we know what the epitope is that's doing the cross-reaction?
In the NS1, I think there is a cross-reacting with the PDI protein present on the surface of
PDI. You mentioned that yesterday.
Yes.
PDI. What is PDI? Platelet protein.
Disulfide isomerase.
Okay, right.
PDI, and is present in platelet.
Yeah, platelets, right.
Platelets on the surface. And this cross-reaction induces inhibition of aggregation.
Pretty complicated.
Yes, it's complicated.
So next week you're organizing a meeting on dengue, right?
Yes, in Cancun.
Cancun. Really nice place, right?
Yes, nice place. And I hope it will be a nice meeting.
It'll be great. It's from the people you have said are going. Michael Diamond.
Richard Kuhn, Eva Harris, Andrea Gamarnik,
you.
Laura White, Anna.
From New York.
Ana Fernandez.
Yep.
Ana Fernandez.
It's a great
Scott
meeting.
Halstead, Susie Clicks. A big meeting.
I would like to go and do a TWiV with some of them.
Yes, it could be great.
It would be great.
You can stay here.
Great.
What?
You can stay here.
Only one more week.
Stay another week, yeah.
Yeah.
Then I will for sure get sick.
No.
Well, some caliciviruses.
You know, someone said I'm traveling so much that maybe I won't get sick this time.
Yes, I told you yesterday.
Yeah.
Because
You
probably
did, yeah.
you have made enough antibodies.
Yeah,
against
could be. Well, I'm fine
pathogens.
so far. So I could talk about dengue forever, but I want to talk about the Norwalk, Connecticut viruses, right?
No, it's not Connecticut.
Dick Pommier once said,
Yeah.
and then we had someone write.
I don't like to correct him because he's a great guy, but it was not Norwalk, Connecticut.
Yeah, Norwalk, Ohio.
Yeah.
So that was the first outbreak of this gastrointestinal disease.
Yes.
And it turned out to be caused by a new kind of virus. And that's what you work on, these.
Yeah.
So what are they called now?
Well,
Calices?
the family is a Caliciviridae family. So it is formed by 4, almost 5 genera.
Right.
So 2 of the genera, sapoviruses and noroviruses, cause disease in humans.
I remember when you were thinking of working on these, you were emailing me, and I said, I'm thinking of working on these. What do you think of that? Yeah. It was a long time ago.
Yeah.
So is this a problem in Mexico? Medical problem?
It's a worldwide problem. So in the United States, just in 1 year, you have like 23 million cases, 50,000 hospitalizations, and 300 deaths.
This is the U.S., right?
This is the U.S.,
Mexico.
But now it's getting, I think, worse. And in Mexico, we don't know yet. We just know that antibodies are acquired at very early ages. Antibodies
to the noroviruses,
to the norovirus. Yeah, to norovirus.
So the sapo is the other genus?
Yes, sapoviruses also cause disease, but it's mild.
I see. Okay. And so these are all RNA viruses, right?
Positive strand RNA viruses.
With no membrane, right?
No membrane. Icosahedral capsid.
Right. About the same size as poliovirus.
The same size as poliovirus, but they look like a spheric virus, but like small round structure virus.
Yeah. So in Norwalk, Ohio, this was the first outbreak, and that was shown to be these new kind of viruses. Is that right?
Yeah.
Or was it many years later shown? I don't remember.
No, I think that there are some reports that they just talk about this vomiting winter disease.
Winter vomiting disease.
Winter vomiting
Wonderful
disease.
name.
Yeah, exactly. That was before that. But then in 1968 there was the outbreak in this school, And by, I think, 2 years after that, they just got for the first time, a virus that causes gastroenteritis. So then they identified rotavirus and then other viruses such as astroviruses and
these are all different
adenoviruses.
viruses. Yeah. So the Norwalk viruses, you get infected at an early age. It seems, as you said, the seropositivity is very early on in a majority of the population.
Yeah, in developed countries, you get the antibodies after that age. But in developing countries, it looks like the antibodies are, you get the antibodies in the early ages.
So in Mexico, everyone would be infected early on.
Yeah.
And so when you're infected earlier, is it not likely to get disease or?
No, there is not a long-term immune response. So you can get the infection
over and over.
Over and over.
Is there just one serotype or multiple?
No, there are multiple genotypes.
Okay.
There are variants of these genotypes.
So if you can be infected with multiple genotypes,
yes.
You don't have protection. So when people come to Mexico and get sick, this is not what's causing it.
No, well, mostly it's caused by an E. coli.
Okay.
This enterotoxigenic. Yeah. But there is a study that was performed, I think, in Houston, in a group of Houston, DuPont, I think, that they just made some research in traveling diarrhea from American people coming to Mexico and Guatemala, and they have seen that noroviruses also cause the travelers' diarrhea. And if you got both bacteria and virus, the case is much worse. Yeah.
Wow. So what is how long does a norovirus gastroenteritis last?
Yeah, like 3 or 4 days. Yeah.
And you feel very bad.
You feel very bad. Yeah.
And then usually you recover.
You recover.
Does it cause death ever?
Yes, it causes death. Well, the two populations that are quite sensible to this is the youngest, the elder, and the immunocompromised people.
Sure. So the babies they can lose a lot of fluids, and unless you replace them, they die. Yeah. How do you get it?
It's a fecal oral route, so mainly by eating some contaminated food, but it is also important the person-to-person transmission.
So it often happens on a cruise ship, right?
Yeah. Well, it has been, there are a lot of stories about cruise ship infections, but it is also, the outbreaks also are caused in semi-closed places like hotels, elderly nurseries, is it called?
Nursing homes, sure.
Nursing homes or schools
too, right? And now that we have more daycare centers where kids are contaminating each other.
And this is an important pathogen, intra-hospital pathogen.
So the hospital workers can be shedding it and they have their hands contaminated, right? And they can spread it to the patients. And so is it a seasonal disease typically?
Typically it's a seasonal disease and it's during winters, but
winter vomiting.
Yeah, winter vomiting. Yeah, but I think that there is much work. We have to do much more work in the developing countries to see if this is the truth, because it has been associated with winter, but there are lots of reports that said that it's not just during winter.
Yeah.
But mainly. Okay.
So these are not easy to grow in culture, right?
No, it's complicated.
So the human viruses do not grow. If you take stool of someone with the disease and try and put it in tissue culture, it won't grow.
It won't grow. So there have been some efforts doing that. So you can say now that it's not conveniently cultured. But
I see.
Yeah,
good.
But I think that lots of real good researchers are working hard trying to get a culture system.
Are you trying to do that?
No, I'm not doing that.
So the genome was cloned, and obviously before we could even grow the virus, it was just cloned from stool, I guess?
Yes.
And so we know the sequence.
Yes.
It's how big?
It's like poliovirus. It's like 7.5 nucleotides.
Now I understand why you like to work on it. And it's positive stranded.
It's positive,
And it-
single-stranded.
And it has a VPG at the 5'
It has a
end.
VPG in the 5'
Just like the
end, yes.
picornas. Are there homologies between the genomes of picornas and...
Yes, the
Calices?
open reading frame 1 that encodes the non-structural proteins are quite similar to the proteins encoded by poliovirus.
So maybe they had a common ancestor years ago.
Well, I don't know. I don't know. But BPG in the case of caliciviruses it mainly works during translation of the virus.
That's right. It helps recruit the ribosome, right?
Yeah.
So there's no iris in calici
No,
genome.
there's no iris. But of course, they have lots of stem loop structures. Well, not lots, but they have stem loop structures that may be regulating all this replication and translation.
It's very interesting. So I'm asking you a lot of questions about the biology. We actually never talked about it on TWiV at all, except to make the mistake on the city of origin.
Yes, exactly.
So the human strains cannot be propagated, but there are some animal strains that can be?
Yeah, well, you can propagate some porcine viruses in notobiotic pigs. And
not cell culture, though.
Yeah. So,
in cell culture, too, you can grow them? And the pigs get sick from these viruses or no?
Yes, I think so.
And what about cats?
Okay, that I was about.
Do they get sick?
Yeah. It's not the same genera. So the cats are infected with the feline calicivirus, which is a respiratory virus, but it has been the best animal model until recently. I think that in 203.
2003?
2003. I'm so sorry,
A long time ago.
In 2003, it was discovered a murine norovirus, which is a norovirus.
Oh, what's his name in St. Louis, right?
Yes.
Did he discover that?
Yeah, Virgin.
Skip Virgin.
Skip Virgin.
Yeah.
Yeah. In his lab.
In his mouse colony, I think, right?
Yes.
Wow,
Yes.
right.
They didn't work with the viruses at all, I think. So they were most working with immunology. So they discovered the virus there, and it has become the best model, I think, because it belongs to the same genera.
Yeah.
And
you can grow it in culture, right?
You can grow it in culture. You can propagate in the mice. And other thing is that it is also a fecal oral route.
Right. Do they get sick, the mice, when you feed them?
Not the
murine neuro.
Well, the mice does have an infection because they shed the viruses.
Yeah.
But the ones that get very sick and die are those stat negative mice.
Without part of the immune response.
Yes.
The stats which transduce the interferon signal into the nucleus and have the cells make ISGs. So I don't know if you know anything about this, but I read that there are many marine caliciviruses, like the whales, and they can make people sick. Whales excrete huge amounts, and people who are swimming in waters that are contaminated can have rashes, and that sort of thing. And also, that's true? That's correct?
Yeah.
There are many infections near marine parks. So, where they have these whales jumping out of the water and splashing people. These people often are infected by marine caliciviruses that are excreted by these animals. Do you know anything? Have you ever heard that?
Yeah. I don't know. I have read a report that some calicivirus from animals was causing a rash in a man, but was working with the virus. But as soon as I know, it's not proved that so the transmission could be also from an animal to a man.
Right. So what aspect of the biology are you interested in? What do you work on in your lab?
Well, in the dark side.
You're on the dark side? You're not in the light side at all?
No. We're just interested in two main stuffs. So one is if it is a real important pathogen in the Mexican population, which it should be. Worldwide, So
probably.
And we know we don't have exact the exact information about developing countries, but in developing countries it should be worse than in developed countries because of the conditions and
Sure. And not enough medical care and
There
can't
were a
give...
lot of things.
Yeah.
Uh-huh.
So how are you doing that? You're doing epidemiology? You're looking at serum?
A little bit. I don't know anything about epidemiology, but I am trying doing something, because I think it's important for our country to know.
Yeah. Can you get serum samples and look for antibodies?
Yes. What we did was to work with 1,100 samples of kids that were to the hospital because they had gastroenteritis and they have to go to the hospital
Mexico
because
City?
it was acute gastroenteritis. Yes, and we choose the groups that have done that, look for enteropathogens, and all those samples that were negative to bacteria, parasite, a lot of enteric parasites, bacteria and whatever, and we just test that, and like 5.3% of these samples were positive to noroviruses. So this was a relevant thing because they had to be hospitalized or go to the hospital because it was a heart disease and it was not caused by other pathogens but noroviruses. So this is a work that suggests that noroviruses could be an important cause of disease in children in Mexico.
Are you going to continue these kinds of studies, do more of them?
Yeah, I would like to.
Could also sequence these viruses and see if there are different strains or whatever in the country.
Yeah.
And look in other cities in Mexico as well.
Yes.
But this is not the dark side. This is the white side.
Yeah, this is the white side, exactly. I was, because I like to do that and I think it's important.
Sure, yeah.
But what I like the most is working with the molecular biology of the virus.
So what's the dark side of your
The dark side is not too much about that, but we would like to see how the host cell proteins are involved in viral replication. So we just look for in the beginning of our work, we just see that the virus has three open reading frames, and upstream of these open reading frames and within the open reading frame there are lots of stem loop structures. So we first seeing if these structures were targets for viral protein attachment, and we just identified like in the human norovirus like PTB, LA, or some proteins that are involved in translation and replication of poliovirus and other viruses. So what we did were some experiments and competing the untranslated region of poliovirus. We see that almost all proteins were compete. So by that time we just identified PTB, LA, PAC, PCBP, PABP, and that.
Binding to which part of the genome?
5' and the 3' end of the norovirus RNA.
Okay.
Yeah. Then we got the feline calicivirus, which is a great model, and recently the murine norovirus. So we are trying to do some of this in order to see if they have relevant roles. So it is not much done on that, but several groups have identified PTB as an important protein for inhibiting translation and letting virus to replicate its RNA.
Right.
And now we're working with the nucleolin, which it seems like it's important for feline calicivirus replication. And we're just using also the murine norovirus model, just to see the differences and the similarities among different genera of the family.
So basically you use the human RNA to identify proteins that interact, but since you can't do a cell culture system, then you go to the animal models and see if that binding is important.
Yeah.
And until maybe one day someone makes an infectious clone of the human strains, and then you can do it there as well.
So I have the feeling that pretty soon we will have that. Yeah. There have been a lot of improvement in the murine norovirus genetic reverse systems, and hopefully we will have a human soon.
Well, for a long time, Hep C could not be propagated, and then at some point. So I think all viruses will eventually be propagated. There are some new rhinoviruses, the rhino Cs. They're just discovered by sequencing, but they cannot be propagated, but I'm sure they could be. You just have to find the right
The right
conditions,
cell,
the cell,
or condition.
the sequence.
there are great people working on that. So I think
Like you. Just laughing. You're not denying. She didn't deny it. See, but you're not trying to figure out how to grow the viruses in culture, right?
not yet, not for the moment.
You will let someone do it.
Yeah, I'm letting somebody
that's
else
fine.
doing that.
It's okay. You have a small group,
I have a small group. Yeah.
and some PhD students.
Yes.
You like it?
Yes.
You love it?
Yes.
You said today in the car, you said to me, I love viruses.
Yes, I love viruses.
Wonderful. Te amo. How do you say it in Spanish? Te amo. No, not te amo.
No,
That's
te
I
amo.
love you. How do you say I love you?
Amamos a los virus.
Amamos a los virus.
Yes.
Well, that's the motto of TWiV, right?
Yeah.
Yes.
Amamos a los virus.
You know, which is really amazing for me is how much information you can get from them. And it's amazing what they do. So if you see what particular things they move, it's just like somebody said that nobody in these times get impressed with anything. They should look at viruses.
It's true. They're not impressed because they don't know, I think.
I think, well, maybe, or because we are just, you have lots of information now. Nobody gets impressed. I remember when I was in your lab that you get the papers by computer. So you didn't have to go to the library. And I say, my God, no. And now it's
routine.
There's a generation exactly. But I think that viruses are a great example of how impressed you can get knowing them.
It's great. No, they're not. Great. I mean, it's interesting for understanding disease, but also, as you said, they let you understand the cell.
And they can do a lot of things with really few amount of genes.
Yeah. Some
because
viruses
they only have 7 or 10 or 12
some have one
genes.
or two. Some have one or two genes.
Only.
Some viruses, and they can do amazing things.
Yes.
They have learned to steal from the cell, right? And take what they need and just supply one protein that modifies an important protein of the cell, and it's just
they are like a Trojan horse.
Trojan horse.
Yes. Yeah. Much more than a zombie.
Yeah. So do you think viruses are living?
Oh, nice question. Inside the cell, maybe.
Ah, interesting. How about the virion particle, though?
No, it's just like a baby, just about being the liver,
Yeah, I'm beginning to think that. What do you think? Do you think viruses are living?
Inside the cell.
Inside the cell.
Of course.
But not as the particle?
No,
as a particle,
not
like a resting living.
Well, it's like a seed of a tree
like a
or
seed
a plant,
or
right?
Spore or something.
Can be living, but you have to put it somewhere where it can. So I'm beginning to see this distinction. The particle is not living, but once it gets in the cell, then the cell is alive, obviously, and then the virus can multiply and evolve and do all these things that we associate with life.
Yeah.
And avoid immune response.
Yes.
Which is amazing.
Every virus would be eliminated if they didn't do it, right? So your viruses, caliciviruses, we don't know. Do we know evasion mechanisms there? I think there are some, right? I think Virgin has done some of that.
Yes.
But we don't know the whole story.
No.
And for dengue, I think I met someone here who was working on that, trying to understand the interaction of dengue with the innate system. There are some people who are working on that as well.
Yeah.
But it's a big deal.
It's a big deal.
It's really interesting. All right. Well, I think we have to move on to other parts of TWiV now. So we're not going to do any email today, but I understand that Lorena has a pick of the week. I do too.
Sure.
You don't have to have one. It's okay. What do you have for us?
My pick of the week is the calicivirus Molecular and Cellular Virology book, which is edited by Grant S. Hansman, Shi Jason Yang, and Kim G. Green.
Recently published?
Recently published in April of this 2010. Sorry.
It's okay.
Yes. And it is. It contains a lot of reviews about certain important things and the state-of-the-art things about different topics on noroviruses, like norovirus epidemiology, and calicivirus environmental contamination. Some others like genome organization, proteolytic cleavage, and viral proteins, which is a really nice topic. Virus-host interaction and cellular receptors of caliciviruses. Reverse genetic and replicon systems, which has been really helpful to understand these viruses. One chapter of feline calicivirus, which has been a nice model to study caliciviruses. Murine norovirus pathogenesis and immunity, and murine norovirus translation, replication, and reverse genetics. And some other calicivirus, which is the rabbit hemorrhagic disease virus and other lactoviruses.
Wonderful. Great pick. Nice book. People can buy it on Amazon.com, right?
Yes.
That's where you bought it.
Yeah.
Wonderful. That's great. I didn't know this book existed.
Yeah.
Are there meetings for caliciviruses too?
Yes, it has been. The meeting was like 1 month ago.
Did you go?
No, I couldn't. It was a pity, but it was in Chile, and as I know, there were great works. Yeah, it's each 3 years, so it seems like the following one is going to be in 3 years in China.
Oh.
So I would be there.
You'd like to go there, huh?
Yeah, sure.
Wow, that's great. Well, thank you very much. You want to pick something?
No.
I'm going to pick this.
I
have a pick. I have a pick.
Okay,
which I
take
saw.
it.
It's a paper that is stuck to your board here in your office, and I'm going to pick that because it's called Insights into Dengue Virus Genome Replication, and there are 3 authors: Sofia Alcaraz Estrada, Martha Jacopicio Monroy, and Rosa Maria Delangel, who I don't know. Is that you?
Yes, I think so.
This is a review article in Future Virology, published 2010. Actually, this is new, right?
Yes, it's really new.
Is it out yet? Not yet?
Yes, it's out.
So we have a wonderful summary of dengue virus RNA replication by none other than Rosa. So that's my pick of the week. Is this an open access journal? Do you know?
No, it's not an open access journal. But I think we tried to put good information in this review, good information about dengue virus replication, and I think include some important topics that have to be considered for the next year.
Do you have a PDF of this?
Yes. Do you want it?
Yeah, I'll get it.
Okay.
I'll send you an email. I like at the end it has an executive summary.
Yes.
This is for busy people.
Yes.
All the article.
That's great.
Epidemiology, structure, replication, cis and transacting elements in the replication complex. So that's my pick of the week.
Okay, thank you.
People check it out.
Thank you for the promotion.
Of course, and thank you for having me. Oops, I ruined your thing there. That will do it for another TWiV, and of course you can always find TWiV on iTunes, at the Zune Marketplace, or at TWiV.tv, where you can play the episodes or download them to your computer or iPod. You can also listen to TWiV on your iPhone or iPad using the Microbe World app. It streams episodes. Check it out at microbeworld.org. And of course, as always, send us your questions and comments to TWiV at TWiV.tv. And if you have any questions about dengue or caliciviruses, we'll pass them on to Rosa and Lorena.
Okay. Thank you so much.
For what?
For coming, for visiting us.
I've been talking to Rosa Maria, who is a professor here at Sinvestav.
Yes, in the Infectomics and Molecular Pathogenesis Department.
Excellent. Thank you for talking with us. I appreciate your time. And Lorena is also a professor here at Sinvestav. Same department?
Same department.
Thanks for joining us. Do you have a website?
Yes.
In the Sinvestav? Yes.
Institution, but you have your own websites as well?
No.
You should.
We should.
You should have a website, a blog, a podcast.
Yes.
Twitter, Facebook, everything.
And TWiV in Spanish.
TWiV in Spanish, yeah.
Esta semana en Virologia.
Wonderful. That's great.
It sounds great.
It does sound great. Well, I'll put a link here to the Sinvestav website, but someday you should make your own pages. You know, Dixon de Palmier has verticalfarm.com. And Alan Dove has alandove.com. And I'm Vincent Racaniello. I have virology.ws. where you can find out all about me. So Rosa, can you close out the show?
Ok. Ustedes han escuchado esta semana en Virología. Gracias por acompañarnos.
Another TWiV is viral.